right, ladies and gentlemen, welcome back. We're going to continue talking about the genetics of bacteria and archaea, and today we're going to talk about this concept of genetic recombination. And really, all that means is we're recombining DNA from two sources. So, combination, right? Recombining DNA. Um, and this is really going to be the major contributing factor to the diversity that we see among these bacterial and archaea species. And to be fair, genetic recombination in general is where we get the greatest diversity of organisms across the board. When we look at things like us or mammals, higher order organisms, most of that genetic recombination is coming as a result of sexual reproduction. We're recombining genetics from two different sources, those two different sources being the parents. Because prokaryotes are asexually reproducing, their genetic recombination isn't going to be able to come from their reproduction. Instead, their prokaryotic DNA coming from different individuals is going to be brought together in one of three processes that they are capable of doing that is sort of unique to this um, set of organisms. So we're going to talk through transformation, transduction, and conjugation. And really what we're looking at here is horizontal gene transfer, where I'm moving genes among individuals of different species and also within the same generational time, as opposed to vertical transfer, where we're talking from parent to offspring. Okay. So if I look at transformation, you guys are going to take a look at doing this in the lab. So transformation, your prokaryotic cells are going to pick up fragments of DNA that are free floating in their environment and actually incorporate it into their DNA. Those fragments of DNA can come from any number of sources. Um, Most of the time it's from a bacterial cell that kind of has died and just sort of left its genetic material hanging out in the environment. But or it can be things like plasmids where they'll pull them out of the environment and take them in as well. So when you guys do the transformation experiment in class, we're going to be doing a plasmid transformation. So bacterial cells take up that genetic information out of their environment, incorporate it, and utilize it. This does take energy, but if that new set of DNA is useful to them, they'll hang on to it. If not, they'll kind of just spew it back into the environment again and move on. Transduction happens as a result of infection. So bacterial cells can actually get viruses just like we do. What we talk about are these bacteriophages, these guys. These are specific viruses, groups of viruses that infect bacteria only. And what ends up happening is in a general viral infection, right, that phage comes in, attaches, injects its genetic information, chops up its host's DNA so that the host can no longer do its job, um, take over the host machinery, reproduce itself, burst out, and move on. But occasionally, what ends up happening in the process of transduction is instead of that virus accurately packaging its own genetic information, it accidentally packages some of the host's DNA instead. So when that transformed, or that transduced, I should say, phage goes down to infect its next host, when it injects that genetic information, it's not injecting viral information, so that new host isn't going to get a viral infection. So the DNA isn't going to get chopped up because there's no instructions for enzymes to break up the host's DNA. And so instead, it can incorporate that genetic information into that new host. And if that happens to give it some new traits that it didn't have before, well, now it's got some new abilities. So transformation, it's pulling information out of its environment, whether it's just free-floating strands of DNA, or a lot of times these are plasmids, even though the diagram here isn't showing it in that respect. Or it can happen as a result of a viral infection. Conjugation is the only one that's going to happen cell to cell, and this is really the closest thing that prokaryotes do to what we would think about as sex, um, because they do create what we call a mating bridge. And those mating bridges are those sex pili, where one bacterial cell has the ability genetically to produce that sex pilus. It's often on a plasmid, that information to produce that sex pilus. And so that bacterial cell will create that mating bridge, create that sex pilus attached to a bacterial cell that does not have that ability, replicate that plasmid, and then send that plasmid through that sex pilus over to that next cell. And now that new cell also has the ability to continue down the line doing this process of conjugation. So that piece of DNA is what we call an F factor. And that F factor is the information to produce that mating bridge, produce that sex pilus. So a F positive cell is going to be your donor. It's going to be the one that has the information in order to do that. Your F negative cells would be the recipients, the ones who do not have them. And this is showing the process if that F factor is on a plasmid. So we send that plasmid down the line, and that allows them to then become two F positive cells at the end, because they now both contain that plasmid that contains the information to make those sex pilus. And more often than not, that plasmid also contains other genes beyond just the ability to produce the pili. If it's part of the chromosome instead, then they are what we call HFR cells. Okay, so still a positive cell in the sense that it has the F factor, but it's now part of its main chromosome instead. And so it's going to duplicate its entire chromosome and send that over to an F negative cell. And what ends up happening then is there are enzymes and things present that chop up the additional DNA that isn't part of that F factor and incorporate in that ability to produce the sex pilus. So they end up being these recombinant F negative cells. One of the biggest things with recombination, whether it happens from transduction, transformation, or even conjugation, are when those plasmids that they may get in any one of those forms comes from these R plasmids. R plasmids are antibiotic resistant genes. Let me take this a step back for a second. Keep in mind that plasmids often contain more than a single trait. Okay, so depending on the context we're talking about them at that moment, they may get some slightly different designations depending on the context. So just keep that in mind. So our plasmids, somewhere in the set of genes that are coming on that plasmid contains an antibiotic resistant gene. It can contain multiple antibiotic resistant genes, or it can contain resistance to a specific antibiotic. The point is that those antibiotics exist in nature. All right, we're generally not synthesizing antibiotics from nowhere. Although we're creating them in lab settings, they have a natural origin. So we look at a population, and this kind of goes back to evolution. That population has been in an evolutionary arms race with the host that produces those antibiotics. Mostly it's bacterial cells going after things like plants and fungi and that sort of thing. And they have been co-evolving over time, where these bacteria are in the environment, tapping into that host is producing those antibiotics as a way to get rid of them. We now use them and have run into the same problems. We're accelerating that evolution. So I get a population where the vast majority of the species, individuals in that species, in that population, are going to be susceptible or sensitive to those antibiotics, but you're going to have variation in there. a range of individuals who are more sensitive versus more resistant. Well, I go ahead and I use that antibiotic, and that antibiotic kills off most of the population if it's used correctly, but it's going to leave some individuals in there. Next generation comes around, and I now have a more resistant population because I killed off most of the sensitive ones the first time. I use that antibiotic again, and now I'm going to kill off a few more of those sensitive ones, leaving more of the resistant ones to reproduce, and now they're even more resistant on down the line. And so we're running into issues where antibiotics are being less and less effective quicker and quicker and quicker because we're using them so widespread. And we're not necessarily, at least historically, having been using them effectively. You can't use an antibiotic to treat a viral infection. We didn't know that initially, and so we were just out there prescribing them at random. Not every antibiotic is as effective against certain species. So again, we're contributing to resistance. If you don't finish the course of antibiotics, then you leave the potential for more resistant bacteria to hang out, more antibiotic resistance. If we don't use the right dosage, so on and so forth. So although medically they are certainly wonderful drugs, they have allowed us to do a lot of things. The evolution that's occurring, even under good usage, mind you, is becoming a more increasing problem. And because these bacteria are capable of sharing that genetic information, sometimes it doesn't even necessarily take a huge use of antibiotics and generational times to get that resistance to evolve. But it's possible that this resistant bacteria is also capable of conjugation, and so now it shares that resistance with this guy. Or something else causes that bacteria to die in the environment, and so those plasmids are now free floating, and this guy happens to take it up. So we're accelerating that natural selection by using those antibiotics, where before it may have been a little bit more of it having to do transformation or transduction or conjugation in order to spread that resistance around. Now we're compounding it by allowing natural selection to happen even quicker on top of all of those other processes that are also capable of spreading that genetic information around.